Welcome, everybody. I'm really excited to be here. For those of you who don't know me, I'm Jenna Monroy. I'm at the Claremont Colleges. And so I am in sunny California, but I know many of you are tuning in from other places. So it's morning for me. It might be afternoon for all of you. I'm going to go ahead and share my screen, but keep in mind that I'm going to end up sharing a screen and then unsharing and things like that. Hopefully I'm sharing the right thing. Okay. So me. Me first, hold on. Um, all right. Can everybody see the right screen? It should be a big screen that just says, Welcome to the IMSI Summer Workshop #1. Let me just go back first. So has everybody signed in? And so that, you get sign into the workshop and the reflection. I'm just going to put that up there for right now. You can scan it and make sure everybody's in where they need to be.
We actually have your presenter mode right now, just as a
Ah, wrong one. Hold on. Let me see if I can do this again. Okay, so let's stop sharing. You can tell I'm not really a Zoom person. In fact, I really hate Zoom. Just so you guys all know that. Okay, I'm gonna try this again. So we'll do that one. And then if I go here, do you see the right screen this time?
yes.
Okay. Lovely. Okay. Um, anyway, again, I'm Jenna Monroy, and like all of you, I'm interested in how animals move and how muscles function. And I was thinking about, what to do for the first workshop of the summer. And I thought it would be best that we actually review the basic principles of muscle function before you guys dive into your research and learn about all these other research projects that everyone else is doing. And so we are at different stages of our career. Some of you know a lot about muscle, and some of you, this is your first time really thinking about muscle. So, what I encourage you guys to do today is for those of you that are further along in your career, I encourage you to act as a mentor today. You might know more than someone else, and so get them up to the same level as you so that we can think about muscle together. The other thing to know is that we cannot cover everything there is to know about muscle in a single workshop. And so the goal of today is really review those general principles of muscle physiology. I'm also going to encourage ourselves and others to think across biological scales, and hopefully we can generate some discussion about aspects of muscle function that are not well understood. Okay, so, you know, like you guys, I'm interested in studying movement. And one of the remarkable things about the muscular system is that it's so highly conserved across animals. The way that they are organized, how force is generated, and even how they respond to length and activation. And so this allows us to identify these sort of fundamental principles and common mechanisms. And it allows us to make broad claims about how muscles function across species. And this is why we can study so many different organisms. I was struck by all the diversity of projects that you all are taking part in this summer. I saw locomotion in tree shrews and K-rats, studying bullfrogs, guinea fowls, and I even saw a project on balance in pregnant women. And it sounds so exciting to learn about how muscles are functioning in all these different organisms. Now, we can also take the alternative approach, right? Is that we have to recognize that muscles are versatile and adaptive. They can function in so many different ways, not only as motors, but also as springs or brakes or struts. And their properties are finely tuned to meet the demands of a particular behavior. And we have to think about those small, subtle differences in muscle, maybe in its structure, or maybe a protein isoform, or its architecture or neural control that can alter and potentially even dramatically alter and animals' performance. And so we really need to consider both perspectives. We need to think about this generalized muscle perspective or this specialized muscle perspective to help us understand how a common biological system can lead to such remarkable functional diversity. Okay, so our goal today is to essentially, we want to be able to explain the fundamental principles of muscle physiology. Think about what you learn in your physiology class, right? And we'll talk a lot about what did we learn in physiology and what are we learning here. I also am going to emphasize that we think about variation across muscles and species. So where are those subtle changes taking place? We're going to evaluate muscle function across biological levels of organization. Many of us work at one level. Maybe we study animal performance, and we don't often think about those molecular mechanisms that are taking place. Alternatively, there's people who study myofibrils, and they never really think about the animal performance. We're going to try to emphasize that all of us work across those scales. And lastly, I'm going to have us try to identify limitations of current theories in muscle function so we can start to brainstorm new ways to measure muscle properties. Okay, so what are we going to do today? We are going to employ what's called a jigsaw method. And so you are going to be given two different groups today. First, you're going to be assigned an expert group. And in that expert group, you guys will learn about one single aspect of muscle function. You will become the expert, and you guys will create a Google slide that kind of represents the main key points of that muscle function. Later on, you will be assigned to a team, and we will have representatives of each expert group in each one of those teams. And then you will tackle more complex problems in your team. All right, I'm going to unshare right now, and I'm going to draw your attention to our Google Doc. Let me see if I can share the right thing. Go here. Okay, so now I'm going to share a Google Doc. Many of you can open it on your own. Okay, let's go back to this. All right, so hopefully you can see my Google Doc. Is that correct? Yes, I got a thing. Okay, so the first document page, if you click on this is essentially what we just talked about. I gave you sort of the overview. This is what we're doing. But then you can see 6 tabs over there, expert 1 through 6, that have different aspects of muscle function. So I'm just going to click on expert 1 just to show you what it looks like. So in this group, they're going to explore muscle and sarcomere organization and structure. And there's a handful of questions there. What is the hierarchical organization of muscle? What is the structure of a muscle sarcomere? How do these structures vary across muscles and species? And what aspects of muscle structure are not well understood? So, given those questions, together you're going to create 1 or 2 Google slides that you're going to present to the rest of the group. Now, keep in mind, we don't have a ton of time. I'm going to try to keep our presentations to like 4 minutes. In that folder, I give you some figures. So I'm giving you something to work with, and I'll show you those here in a minute. So include the figures. You can move them around. You can cut them up. You can choose what aspects you want to focus on. Give some bulleted text of those main points that you want to discuss, and maybe come up with a set of questions. Each person should have a role. So make sure that maybe you have somebody who's the slide organizer, the person who creates the slide, shares it with everybody, and moves things around on the slide. You have people who are researchers. Those are people who might find information that's needed to complete the slide. And then you identify who are going to be the presenters, who are going to be the people who present the slide. Certainly, any and all of you can contribute in that way. So it's up to you how you want to delegate tasks. Notice I give you 2 resources at the bottom. One says sort of the slide. There's actually 2 slides for expert 1. I'll show you where that is here in a second, and a related paper. So I'm going to click on this tab. So this is our folder. Do you guys see the folder now on my screen? And what you can see on there, I like the thumbs up, thank you. Sometimes talking into a black box, you never know what you're doing. we can see our Google Doc in here. We see a handful of papers, and then we see a PowerPoint in there. If you click on the PowerPoint, it opens up in a Google slide. You can see the figures that I have provided for you for each of the expert groups. So we have expert one actually has two slides, and these look like typical figures that you might see in a physiology textbook. And there's a second one for expert one. But expert two only has one slide. And so you can see expert three has one slide. Then we kind of go down. Everybody has one or two slides that they can include in their presentation. All right. So the last thing I want to point out is that I also gave a paper associated with that particular topic. So in the case of muscle and sarcomere structure, you can click on this paper here. Do you see the paper? This is Leroux et al. This is a really cool paper. By no means do I expect anybody to read the paper in like the 15 minutes that you have, but it does have some really cool information. And perhaps for those of you that are a little more advanced, you can look at the introduction, maybe some of the figures that might be relevant to why or how potentially those structures might actually vary across different organisms. And in this case, this paper is about insects and sarcomere structure in insects. So that way you can address some of those questions about how these structures might vary across different organisms. Okay, I'm going to stop sharing for a second. All right. Let me come back to the group. Now, does everybody know what they're going to do? Victoria's going to give you an assignment to one of those expert groups. We're going to take 15 minutes to create our slide. And then for those of you who are faculty, at this point, I would like the students to lead the groups and the faculty can stay here. But let's imagine that some of you get stuck in your group and you need to call upon a lifeline. That is where a faculty member could join a group and they could give some insight about what they might need to put into their slide. All right, are we ready? Let's do it. All right. It was a little quick. I know 15 minutes, you're like, I didn't actually finish everything that I wanted to do. But hopefully you guys have stuff to say. And I'm gonna try to keep us on task. So because we don't have a ton of time. So I'm gonna ask expert group one, have you identified a presenter? And would you like to share your slide, present your slide for us?
I'm the presenter for expert group one. So I will try to share my slide now. Let me get into, okay, share screen. Yes. Do you see my screen now?
Yes.
Okay, great. So we are talking about muscle and sarcomere organization and structure. So here we see a single muscle fiber, which interestingly can span the entire length of a muscle, I believe. And like it's very different from most cells. It can be very long. Um, and the smallest unit of a muscle is a sarcomere. It has the typical length is 2 to 2.5 micron, and this is pretty consistent across organisms, both ranging from insects to elephants, I believe. You can correct me if I'm wrong. Um, and it's got some of the structures in it are described here. So this is called the Z disk. The Z disk has a uh, filament coming out of it, which is I believe, the thin filament. Yeah, that's the green one. And then in the middle, there's a M line. This is called the M line. And then there's another filament called the thick filament coming out of it. Go to the next slide. Yeah, this shows a more detailed view of the sarcomere structure. Z-disc, M-line, thick filament, thin filament. Actually, I'm using my mouse to point. Are you
Mhm.
able to see? Okay, good.
Yes.
Then the M-line, and so this is a close-up of the thick filament. It has these myosin heads, which are also called cross bridges, which attach to the thin filament. So you can see that here. So when the muscle is activated, the cross bridges attach to the thin filament, and they pull on them. And what I've learned is that they actually kind of have a swing phase as well. So they're running on the thin filament as the muscle contracts. Um, Another important structure is this part called titin, which connects the Z disc to the M line. And I believe that is responsible for the passive stiffness of muscle. So when a muscle is not activated and the cross bridges are not engaged, if you stretch it, it's the titin that's providing that elastic um, resistance. Um, that's all the time we had for the purpose of making slides. Uh, so I'll try to describe some of the questions about things we do not know. So the Hill model is good at predicting static muscle function, um, but it doesn't match with a lot of the data we observe in dynamic muscle function. And we believe that the this element, titin, this filament, um, plays a role which we don't fully understand. So that's ongoing research. Um, yeah, that's one of the things we don't know, like how are muscles tuning their stiffness in dynamic conditions. Yeah, that's one of the questions.
Great. Does anyone have any questions? So one thing I was struck by, I don't see any hands, is that sarcomere length can actually vary pretty wildly
Oh.
in invertebrates, and
Mm.
much more so than that of a vertebrate muscle. And I'll throw the question out there. Does anybody know about sort of the variation in sarcomere length, in invertebrate muscle?
I do know about super elongation and obliquely striated muscles. So one of the um, animals that we're working with is bloodworms, and they say that for some obliquely striated muscles, they can elongate to maybe 5 times or 6 times their length.
Wow. And they still maintain force too, right?
Right.
And so that's super impressive. So one of the things to take from this is that while the structure may be really similar, the length can vary. And it does seem like tighten is this ruler, but we can change the length of our thick and thin filaments in ways that maybe vertebrate muscles cannot. And so it's one comparison that you can ask is how that impacts the function of the muscle. Are there any other questions or comments about that? All right, I'm going to keep us moving forward. You can unshare or yeah, and
Yeah,
then we can allow
thank you.
group two tell us something about their their slide.
I can share for our group. Let me just pull up our slides. Uh, all right. So we had excitation contraction coupling, and kind of like questions about how this differs across species, and sort of what are the questions that we still have that we don't really understand about ECC. So, um, it might be important to start with what ECC is. And it's because I don't really know too much about it, but from what I understand, it's basically from the start of an action potential at a muscle cell all the way to the actual mechanical contraction that's happening at the sarcomere. So it's everything that happens in between there. A lot of it is like dealing with molecular kinetics of calcium cycling in and out of the cell, from action potentials. So we kind of start with the depolarization signal here that sends sodium through the cell, depolarizes that cell, and that leads to calcium influx into the cell. Calcium binds to troponin, and then you get cross-bridge cycling, and then it can be reuptaken by this like ryanodine receptor, comes back into the cell, and then you kind of get this on and off of like contraction that occurs. Um, so that's kind of the main idea from what ECC is. It's pretty short time in terms of how long it takes from a depolarizing signal to get to an actual like shortening of the sarcomere. Um, and so kind of the questions that we had was like, how does it affect muscle force? How does it vary across species? And what is not really well understood. So our group was able to look at this a little bit. Kind of the second two questions, more or less, which is how does it differ across organisms? And what are the challenges that people who study ECC still face? So one of the things that we saw was um, variation, basically, and sarcoplasmic reticulum density can influence the frequency at which you're contracting, and variation in the molecular machinery can lead to variation in the ECC kinetics. So if you have a higher density of ryanodine receptors, maybe receptors that have a higher affinity, for calcium, that can all lead to variation in how long ECC can take. Um, and so maybe some of the challenges, that are not all encapsulating, but some of the challenges that we noted was consistency in methodology for identifying calcium concentrations. I think this, because you're working at a pretty small scale, like molecular scale, I think there was a lot of complications early on, but as we've kind of grown technologically wise, there's a lot better like fluoroscopy methods to like identifying influx into the cell and then uptake. But I think there, from what we saw, there was still maybe contention between how different labs are doing it, how you can kind of identify the differences between already present calcium that's just floating within the cell and then actual calcium that is implicated in like actually the mechanical contraction. So I think consistency in methodology was one of the challenges that maybe additional tech can like kind of ground. And then one interesting one that I found was IPCS-derived skeletal myocytes and their ECC. Does it kind of differ from normal skeletal muscle? Because I think there's a kind of therapeutic trend going into like pluripotent derived stem cells. And so trying to understand how skeletal muscle stem cells and their ECC differ from normal stem cells, obviously, was something that hasn't been really answered or fully fledged out. And I think that that is all we had.
That's excellent. I do not know much about the iPSC derived myocytes. Those are quite interesting. And you're
Yeah.
right, like, are they actually the same as what you might find in actual muscles? Does anybody have any questions about that? I think that was great.
Yeah, I have a question. Nice work in the short time frame with the slide. I was just curious, what is the time scale of this whole process from excitation to contraction?
I honestly don't know the exact time scale. I would say like on the order of maybe like milliseconds, but I'm not too sure.
Yeah.
Maybe someone could correct me.
Well, think about like a twitch in a muscle. If you guys work on muscle twitches, they can occur in just, your time to force development can occur in just a few milliseconds. And in some really fast muscles, it can be a few milliseconds. For some, it's more like 20 to 50 milliseconds. So certainly you're releasing calcium into the cell from the sarcoplasmic reticulum. One thing that I might ask too is about, so we often talk about force development and the release of calcium. What do you know about the rate of calcium uptake? And that would be associated with relaxation.
What I at least minorly know is that it's a lot slower than the
Hmm.
rate of force development, maybe like sometimes double. Um, I'm not very sure as to why, though.
Yeah. Does anybody else know?
I think usually it's slower to prevent like a retwitch, of activating the same muscle.
Similar, like thinking about like an action potential that might have some absolute refractory period, potentially. One thing to think about is that, and I think we're going to hear this in a minute, is we'll learn about summation of force in a muscle. Well, relaxation is associated with that uptake back into the sarcoplasmic reticulum, and it's associated with what we call our circa pumps. Circa just stands for sarcoplasmic endoplasmic reticulum calcium ATPase. But it takes a lot of energy. It takes ATP to pump that calcium back into the sarcoplasmic reticulum, and those have their own kinetics. They have their own rate. They're often quite slow, and you can upregulate the number of circuit pumps that are on your SR, but to increase the rate at which you go. But keep in mind that's always a little bit slower. And as you pointed out, is that the frequency of contraction is dependent on how quickly you can increase force, but also how quickly you can decrease it. Yeah. All right, I'm going to keep us moving forward. Let's hear
And
from group 3.
I'm sorry, I just want to add this also. All of these processes are so related to the type of the muscles that we have. I mean, fast twitch or slow twitch
Yes,
muscles.
that is quite correct. Yeah. So all these things vary with what we call sort of fast and slow twitch muscles. Yeah. Thank you. All right, let's move to group 3.
Awesome. So for this part, we kind of divided it up by 3 parts: low frequency, medium frequency, and high frequency. But just in part, you can see here for the 1 time per second, 2 times per second, there's no summation. So just kind of meaning that the muscle completely relaxes between the stimuli. It does show like those individual muscle twitches. And then going over to like the middle part, the summation, just kind of meaning that stimuli does arrive before the muscle can fully relax, causing the contractions to run on top of each other. I know we kind of put here medium frequency, the muscles twitches begin to sum up into incomplete tetanus, and that's kind of here on this part. Just kind of meaning that there's rapid, successive stimulation results in a peak force that the plate is visible with. But you can see there's like maybe minor relaxation ripples. And over here on the high frequency, we kind of just came up with the conclusion that those are kind of high stimulation frequencies, meaning that the individual twitches do begin to kind of blend into one another, kind of making one smooth, sustained contraction. Um, and going over to this one, I think there was somebody else in our group that they found this from a different paper,
Yeah.
but I think they kind of wanted to speak on this one.
Oh, well, this is just adding a little bit to what we already heard. I thought that this figure shows really nicely how these individual twitches may sum up at intermediate range of the stimulus frequency. And if we look at the magnitude of the force developed as a summation of these twitches, in the lower range of the stimulus rate, it is monotonically increasing with the stimulus frequency, and it will saturate at some point after some frequency. Yep.
And then just to kind of answer the questions, how does the frequency of stimulation affect the force output of a muscle? We came up with the idea that the increased stimulation frequency does increase the force output of a muscle. And with higher frequency, twitches overlap. How do these processes vary across muscles and species? Force frequency relationships do change. Different species have different locomotor demands. Some animals do have slow oxidative fibers, and others have fast glycolic fibers that later produce a large amount of force faster, but it is quicker to fatigue. And then what are the limitations of this relationship? The maximum amount of possible force is constrained by muscle properties, which does mean that fused tetanus has a fixed upper bound for force generation. And the frequency of stimulation is also physiologically constrained by the rate at which muscle activation is possible. And then what aspects of muscle activation aren't well understood. Understanding from the article that was given was the translator signal. So we do understand that physical muscle pulling does dictate the sarcomere size, but the internal chemical messengers that translate those mechanical forces into a growth command aren't fully understood. So motor recruitment versus firing rates, kind of how those interact to regulate force production. And that's pretty much what we were able to do within the 15 minutes.
Excellent. Thank you. Here, if you want to unshare for a second, does anybody have any questions about this? So I'm going to ask a little bit about so this is a measurement that you take from a whole muscle. And whereas the previous group talked about molecular mechanisms, can you think of how the excitation contraction coupling that they mentioned in the last group relates to what you presented with the force frequency relationship?
Yeah, I think I can answer that question. So in that first figure that we were looking at, we have sort of a quick up spike and then a slow sort of exponential decrease. And that is coming from that relationship we just talked about, where it takes less time to activate the muscle than it does to deactivate and get that calcium uptake happening. And the fact that there's a longer amount of cooldown period makes it so that when those two things overlap, we get that summation property.
Yeah. So if you had another stimulus come in, more calcium is going to get released, and they're just going to add the force on top of the previous twitch. Excellent. So things that I'm trying to emphasize is thinking across scales there, right? Where we're thinking about the molecular mechanisms that might underlie the muscle property that you're measuring. So in the force frequency relationship, we're measuring it from the muscle, but we kind of have to think about all those molecular mechanisms that are taking place. The other thing that I might ask is, can you think of a limitation of that type of measurement? So you can go in, you can measure these, you stimulate the muscle at a certain frequency and you get this nice force profile, then you increase the frequency and you do it again. What do you know about how those measurements are taken? And is there a limitation when you try to consider an actual animal? Does anybody record EMG?
Yeah, I think that the limitation with the way that we record EMG, at least in my labs, is it's a surface EMG. So we're not measuring a singular muscle fiber. We're getting just a sort of general difference between two rough points, but you aren't able to get fiber level activation information that way.
Yeah, it becomes difficult to compare what you're measuring at your EMG to this whole muscle sort of preparation. And the one thing that I was getting at was sort of, we often just stimulate at a single frequency, say 30 hertz, and then we activate the muscle. And if you notice, those are activated at isometrically. So they're not changing length. And we'll talk a little bit about length here in a minute. And then they change the frequency and let's stimulate it at 100 hertz. But when I'm when the nervous system activates our muscle, it can change things. This is what we mean by rate coding, is that it might increase the frequency and then decrease the frequency, and it's tuned to whatever it is that it's actually doing. And in fact, I study something called doublet potentiation, where sometimes you can actually activate the muscle really rapidly, and then you decrease the frequency. And you can see this in the EMG, and we can try to replicate it in the lab, and it changes the force output of the muscle. So things to consider is some of our measurements are often kind of limited and don't necessarily represent what's actually happening in the muscle, or sorry, in the animal. Okay, we got to keep going more forward. Let's hear from group 4, right? We're in group 4.
I'm going to present the first figure, if I can figure out how to share my screen. Awesome.
Oh.
So the first figure is a pretty generic passive versus active force curve with the passive obviously is more dependent on the elastic structure, so that'll be mostly tighten, and then also the extracellular matrix. And so obviously as you're at these lower lengths, you won't get a lot of force, but then as you stretch it out, those elastic components play a much bigger role, and you have this exponential curve. Whereas the active force curve is more dependent on cross-bridge cycling, so you have this sort of peak, which is often referred to as L naught, so the optimal length of the muscle and the force length relationship. And that'll be the like most active cross bridges. And obviously, if you're at a shorter length or a higher length, you'll have less cross bridges or you'll have too much overlap, which decreases the efficiency of it. And one of the sort of questions we had based on that, so I work in Dr. Monroy's lab and we study mice that have a chunk of their titin cut out. And obviously that would change the force length relationship. And one of the questions is like, how would, if the elastic component like titin, if that was altered, how would the force length relationship change in this graph?
Someone else from that group want to
Yeah,
also.
I can do the next
Yeah.
slide but I'll need, okay, perfect. Okay, so this figure is really interesting because the top figure A is comparing the force length relationship between vertebrates and then invertebrates in B. And some of the key takeaways of this are that the inverted U relationship of the force length curve stays the same. So there's going to be some optimal length. And if you lengthen past that point, it's going to have worse contractile mechanics, and same thing if you shorten it. But there are some interesting points about this, which is that the amount of relative lengthening or shortening is different depending on species. So I think it's the mouse limb, the red line in A, is very interesting because it only shortens to like 0.95 of its relative optimal length. And so that could be indicative of its optimal length being in an already very shortened position. There's not much more shortening it can do, or there's some other limitation there. So there is some variation between species on where that optimal length might be. The way they normalize this graph is a little frustrating to me personally, because I would want the x-axis to be relative overlap of the sarcomere, because right now it's normalized to its optimal length, which is normalizing to optimal force. So your x-axis is normalized to your y-axis, which I think you could see a lot more interesting things if you didn't do that. But you can still see some really interesting trends. And then some of the limitations that you have when trying to explain all contractile mechanics just through force velocity and force length is that when you actually put muscles in more realistic conditions, like Dr. Daly has done some perturbed locomotion studies, making work loops out of those. And there's some inconsistencies between what you would expect to see and what you actually see. And so it opens room for other explanatory factors like the tension inherent in like the extracellular matrix, an active role for titin in contractions, and then different calcium uptake or removal speeds depending on your force length or position on the force velocity curve.
Nice. Thank you. Are there any questions? I'm always struck at how variable. Oh, go ahead. Go ahead.
Well, I have a very naive question about the force
Yeah.
length curve. That coming from really naive perspective, I feel like it is always beneficial to have a wide force length curve. Like it would be nice if you have a wide range of operation where you can produce like significant force. So what would be the reason or benefit of having a shallow curve for an animal? Or is there a cost of having a wide curve?
This is a good question. Does anybody have any thoughts about it?
I have a guess,
Yeah.
which is I don't know any situation where it's better to have a really narrow force length curve, but I think if you have um, tendon properties that allow you to activate muscles in mostly isometric conditions, then it doesn't matter how wide your force length curve is because you're mostly going to be acting at that relative length in isometric conditions.
Yeah, I would agree that there are some muscles that barely change their length, and it's the tendons that do the work. So they can stay mostly isometric, and their force length curves can be quite narrow. But as we pointed out, those invertebrates like the annelids and some of the insects have really large sarcomeres, and their force length curves just keep going for a really long time, and it allows them to maintain force over a really large range of lengths. So I don't know, honestly, the cost of that really long thing. I forget who it was who studies the blood worms. Do you have anything to tell us about it?
Sorry, I was muted. I actually don't know much. I just started the project, but I guess the only thing that I do know is that the super elongation that they have with the broad force length curve, they have a specific purpose. And the PI that I work with, he specifically saw that in squids as well. And they said that they saw it in the tentacles. So the tentacles would stretch out super far for the prey strike. And that was why they had the broad curve.
Yeah. Oh, and if you guys had seen on the last slide, there was the chameleon. And so the chameleon can function at really short lengths on the ascending limb of its force length curve. And that is because they're considered to be super contracting. So it means that they can get really short and still maintain force. In fact, the hypothesis is that the thick filaments actually slide through holes in the Z disc and then interact with the thin filament in the adjacent sarcomere. And while they haven't totally seen all of that, it really is mind-boggling to know that they could get that short and still maintain force in ways that other vertebrates cannot. And it's because they can extend their tongues out, but they have to maintain force even as they contract back into the mouth. So there are a lot of cool little changes that are taking place in these organisms that allow them to either maintain force over a large range of lengths or on one side of the curve, either on the ascending or the descending limb. One other thing to point out, right? And I think you've already mentioned this, is that the force length curve is a static property, right? And you've already mentioned about work loops and other ways to really get at dynamic muscle function, which some of these tests, like the force length, and we'll learn about the force velocity relationship, just really don't capture because you collect those data in these really static ways when the muscle's not changing length. So clearly the force of the muscle is going to change if we stretch it or if we lengthen or if we shorten it. All right, let's move forward. Let's go to group 5.
I'm sorry, I didn't know I had remuted myself. Okay, does that look good for everybody?
Yeah, that looks great.
Okay, so like I said, I didn't have time to look at the graphs, but if somebody in my group can chime in and help me, that'll be great. But anyway, okay, so the force velocity relationship of muscle, if you don't know, so as the faster a muscle has to contract, the less force it is able to produce, basically, is what it says. And this force velocity relationship does vary across muscles and species. It depends on the fiber type, whether it's a slow twitch or a fast twitch fiber, and which I think is shown in this graph down here that I didn't get to really look at. But you can see in these graphs there are, let's see, bat wings, fast twitch muscles of mice and slow twitch muscles of mice. So we'll go back to this, but it also depends on the fiber length, which we already talked about a little bit, and the pennation angle. It also depends on the operating frequency of the muscle. The type of contractions. And so, yes, this varies across species too, as we saw in this figure here. Let's see, let me go back to this. And how does this relate to the swinging cross bridge theory? Well, it depends on the rate of basically ATP cycling rates, the rate of myosin detachment and attachment, and force proportional to the number of cross bridges that are attached simultaneously. But what this doesn't take into account is our history dependent effects. It ignores the role of Titan. And it's based, these force velocity experiments are based on Hill models, these experiments were done under maximal activation, based on isotonic conditions. And as most biological movements typically are under submaximal conditions. And then let's see. I didn't look at this one yet, but let's see.
I made this, or like I explained this plot so I can just talk about this, but basically this plot shows like power as a relationship, like overlaid on top of some arbitrary force velocity graph. And the force velocity plot, like it shows this like the downward trend that you see with pretty much every single other muscle. And then because power is a product of force times velocity, you're like the max power would be somewhere in the middle where both force and velocity is relatively high. And it tapers off, or it decreases a lot at the ends. So that's kind of what this plot shows.
All right. Are there some questions or comments? You guys can unshare your screen so we can see everybody. All right. I think you guys hit the main points. The point is that clearly, it can change. It's focused on shortening, right? And sort of shortening velocity. It doesn't tell us much about lengthening. And as you guys pointed out, it's it's a quasi-static sort of measurement. And so it's under these isotonic conditions, your muscle's maximally activated, which doesn't necessarily represent natural movements. And so hopefully we're getting a theme here that the properties that we measure under these highly controlled conditions, and they don't necessarily represent what's actually happening in an animal. All right, any other comments? Let's move on to group 6.
Our group didn't actually choose a presenter, but I can share my screen. I think we're just gonna work off of, oh, sorry. Just gotta figure out where that is. Share. So yeah, we were just working off of the figure provided in the slide. And we touched on briefly, is my screen shared for everyone?
Yes.
So we were just talking about how the figure shows the different pronation angles, and so we were talking about the difference mainly between what it means to have like a very parallel muscle versus a highly pinnated muscle. And so if there's a very parallel muscle, that means that the muscles kind of run along the length of the tendon and all the fibers are parallel, and it allows for more sarcomeres in series, which means that it can shorten a lot and it can shorten faster. Whereas if you have a lot of parallel muscles, it kind of restricts the speed of contraction of each individual fiber. But that restriction allows for more fibers to be packed into a smaller unit, which allows for more force output since there's just a lot more overlap between the thick and thin filaments. And so we were kind of talking about how there's a trade-off between maximum force output and speed of contraction, which I think the expert group 5 covered nicely. And as for the differences between species, I think I heard like the watch party was discussing that a bit, so I don't know if they want to jump in for that part.
I can chime in for that a bit. I didn't get the chance to fully go over all the figures in the paper, but in figure 4 of the paper, they compare the cross-section area and the length of the muscles. And they looked at multiple different frog species, but broke them up into swimmers, jumpers, and walker hoppers. And what's interesting is for the swimmers, as the length of their muscle increased, so did the cross-section area, and that was the opposite for both the jumpers and walker hoppers, which I think is very interesting and shows how when there's different function needed for the muscles, like swimming for these species, then the structure will differ also.
Nice. Does anybody else want to add to that? I like this because here we're talking about whole muscle structure, and in the previous group, they were talking about sort of muscle properties, and combined, they can impact, the rate at which a muscle can shorten and the force that it can produce. And so again, we need to be thinking about muscle properties across different scales from their tendon muscle organization and architecture, thinking about individual muscle properties like we were talking about fiber types or the rate of myosin attachment and detachment rates, and then thinking about those molecular mechanisms that underlie sort of the release of and uptake of calcium. So we can start to see how we have to address all different levels of biological organization to kind of get at these tuned properties of muscle for whatever behavior that they're trying to perform: hopping, running, jumping, feeding, all these different things. All right. This is great. I hope you guys are learning things, right? It's helping you kind of explain things. Maybe you knew, maybe you got a little better of a chance to understand it. So I, of course, over, budgeted or under budgeted my time, but I want to take the remaining time to kind of tell you a little bit about what I do. You guys have already mentioned a little bit about Titan, and maybe we can discuss as a group now that we're all experts in different to kind of think about the role of Titan in muscle as we've kind of brought it up in different ways. So I'm going to go back and share my screen again. Let's see. So let me go here. Oh, no, I'm going to go here. Hold on. Share. And then let me go here and. Okay. Do you guys see my toad feeding? Yes. Okay. I got this. Okay. So I think in our explanations of these fundamental principles of muscle function, I think we all kind of said, oh, yeah, we can measure these things in muscle. But what we are all struck by is that certain properties that we've measured really don't accurately predict natural dynamic movements. And so we're missing something. Our understanding of muscle function is incomplete. And this story actually started with me when I was a graduate student, when I studied feeding in frogs. And so what you're looking at here is a toad feeding on a cricket. And this is done in a high-speed video. And what I want you to pay attention to is that they open their mouths really fast, which then flips the tongue out of the mouth really fast, kind of like a whip. And so it is those jaw muscles, our depressor mandibular muscles, that contract really fast, really rapidly to open the mouth fast. And when you measure it, you get these enormous power outputs, and they don't have any tendons. And one of the things that really challenged us in our research is, where do they get this enormous high power output? And over the years, you might have thought that this was unique to toads and it was something special. But over the years, many researchers, including yourselves and your mentors, have found many examples of organisms that exhibit power output that far exceeds what we predict as being the maximum, because it's defined by our force velocity relationship, which doesn't take into account viscoelasticity, right? And so we know that muscles are viscoelastic. And as you guys kind of alluding to, we know that titin plays a role in that viscoelasticity. And the other thing that you guys have pointed out is that muscles aren't static. So these properties of force velocity and force length relationships are quite static or quasi-static. We maintain them under these highly controlled conditions. In fact, we get rid of the contributions of viscoelastic elements when we measure those properties. But most muscles undergo lots of length changes. In fact, we often think of our locomotor muscles as undergoing stretch shortening cycles. And we know that muscle force changes with length and activation. And like Carissa mentioned earlier, that we think of history-dependent properties, sort of how is force changing with stretch versus shortening. And there's a large amount of data now that support the function of titin as being a viscoelastic spring in sarcomeres. And so let me just point out titin once again. So as you guys pointed out, this is a picture of a sarcomere. And what we're seeing is actually half a sarcomere. So you can see the Z disc on the far left-hand side, and this red structure in the middle, that's actually the middle of the sarcomere. That's the M line. And the other Z disc would be off the screen on the right. And the green structure represents our thick filament, and the blue structure represents our thin filament. And I want you to pay attention to this yellow structure, which is titin. And so titin interacts with the thin filament at the Z disc, and it runs all the way through the myosin core to the M line, where it interacts with another titin filament. And so it essentially creates this lattice that holds the thick filament relative to the thin filament. So it's responsible for what we call sort of interfilament spacing, which can actually affect how they interact with each other. I am particularly interested in this I-band region because that's the viscoelastic spring. And while you guys don't have to remember all the names, there's like these three parts to it that I'm interested in. We're going to call it an IG domain. We have this little red element that's called the N2A, and then we have something called a PVK. And the things to keep in mind is that the IG is kind of a spring, and the PVK is a spring, and that unique N2A, it's like this unique sequence, this unique structure. All right. So as you guys pointed out earlier, there's lots of evidence to suggest that titin contributes to passive muscle stiffness. That is, when a muscle's passive, there's no calcium in the cell, you can stretch it out and you do get some resistance, and you see this exponential relationship between the length and the force output. And you could do this with an individual titin molecule, and it matches what you see in the muscle. So that's what we're seeing in this graph here. And we can attribute the different parts of titin to our passive curve. This low force straightening, where you have this low increase in force with a change in sarcomere, is associated with the stretch of this tandem IG. So this is kind of a spring, but it's kind of a compliant spring. But then as you continue to stretch a sarcomere out, you would actually see that the force increases exponentially, and that is associated with this PVK domain. And I'm particularly interested in that PVK region. And The other thing that's interesting about Titan is that more and more evidence suggests that Titan contributes to active force. And so it's this tunable spring. It changes its stiffness when calcium floods the cell. So imagine that your cell gets activated. You guys just talked about at ECC. We see a release of calcium into the cell. And not only does it activate and expose our actin binding sites, but it actually facilitates the binding of this N2A region to the actin filament. And what it does is that it eliminates these tandem IGs, and it engages only that PVK domain. So what we're doing is we're engaging that PVK domain at a shorter length, and that allows us to have this stiffer Titan stiffness when a muscle is activated. And now you can see that this activated Titan could explain sort of how the stretch of this spring could potentially provide extra force and stretched muscles, and perhaps even in power amplification, like what we saw in our toad muscles. So you got a spring in there, you could store elastic energy, you could release that energy when you do things like open your mouth really fast and project your tongue. Okay, the other thing that I'm interested in, so let's say Titan's contributing to passive force, it's contributing to active force. Titan varies across different muscles and species. We know now that it is at least, so I'm going to talk about vertebrates specifically right now, but there's some cool questions in invertebrates. But in vertebrates, there's one single Titan gene, and that Titan gene can have various different exons be spliced out. So through that alternative splicing, you can have muscles that express long Titan isoforms. And when you have a long isoform, you're actually passively compliant because you're a longer spring. But you can have other Titan isoforms that are shorter and then they are stiffer. And so what you're seeing here is the passive force of our soleus muscle in the leg. It has a really long Titan isoform. It has all those exons in there, and so it has low passive force. But when you cut them all out like that in the psoas, or a lot of them out, now you have higher passive stiffness. So we can see that we can tune just the Titan stiffness in a muscle depending on what these different muscles are doing. And I can tell you that the PVK region in particular is where all that splicing is taking place. And I don't have a lot of time to explain all of this, but the point is that this diagram below can show you which exons are being expressed in each of the different muscles. And I did a project with a student in 2019, and what we did is we took the DNA and the sequence of the Titan gene from lots of different mammals. And I know you can't really see this slide, but I want you to see is just the variability. And so what we're looking at is a phylogeny of mammals from, you've got your platypus and your marsupials at the top, and then various different placental mammals. And what you're seeing on the right is all these little pink and green stripes. That is the exon arrangement of your PVK region. And what I want you to just take note of is just they're not the same. The number of exons and even sort of the color, the pink and the green kind of indicates how much P's, E's, V's, and K's are in there. So we can think about sort of the composition of the PVK as well as the length of the PVK and how those things might differ across different organisms. I can zoom in just on one piece so you can see a little better. So this includes our platypus at the top down to our apes at the bottom. Again, what I want you to notice is that the number changes, how the color changes, and how similar it varies across the different organisms. Well, those exons that are close to the N terminus, we're calling that PVKN, are more similar. And so you can see just down, if you look down that region, you can see that they're relatively the same. But then you look at the PVKC, the other side of the Titan, and you can see it's highly variable. And so there's lots of different variation in our PVKC region. Now, this is just the gene itself. Keep in mind, it still can undergo a lot of alternative splicing in one muscle compared to another muscle. All it tells us is how much variability you might see just across the organisms. So I am particularly interested in how Titan isoform expression can impact, various different animal performance and muscle function. So, one example that's really great in the literature is this paper by Manny Azizi, which maybe some of you are working with him this summer, where this was a 2014 paper where he looked at the function of the plantaris muscle in the leg and the anconeus muscle in the forelimb. And the plantaris muscle, it's responsible for powering the hop in the toad. It shortens during contraction, it produces mechanical energy, it produces power so you can jump. But the anconeus, it undergoes active lengthening. It breaks as the animal lands on the ground. So the plantaris acts as a motor, whereas our anconeus acts as a brake. So we see two muscles in the same organism that have very different functions, and one lengthens during contraction while the other one shortens. We also know the difference in their passive force. So they showed that the plantaris muscle is quite compliant and compared to the anconeus, which is really stiff. And so this begs all kinds of questions about Titan, where just based on the passive properties, we might say, does the plantaris muscle express a longer, more compliant Titan isoform compared to the anconeus? And then we could even ask, is that in the PVK region? Is it in that IG domain? We can also ask, how does active stiffness compare in these two organisms? And so this has to do with that N2A region and the location of that N2A. Are we actually seeing the same increase in stiffness with activation? Are we seeing something different between these muscles that differ in their function? So my goal here is to get you guys thinking about whatever project that you're working on as how Titan might be playing a role and whether or not its properties and what isoform you might be expressing is tuned to the behavior that you're interested in. Okay, so I know that we've kind of gone long here today, but hopefully what you're seeing is that we're kind of missing these measurements of muscle and Titan viscoelasticity from what we've traditionally learned in our physiology classes, right? And so we need to start thinking about variation across muscles in the same way that you guys discussed today, but also when we think about Titan isoforms, and that these variation in Titan isoform could be matched to the behavioral demands of an organism. Okay, so I'm gonna unshare again, right? Because we only have a few minutes. And so maybe I'll open this up to discussion. Ideally, we were going to have you guys go into breakout rooms and come up with your own questions about Titan and muscle. But maybe I'm just opening it up for discussion to you guys. What questions do you have about this?
I have a question about what you presented regarding the Titan isoforms. Could you maybe if you could share a slide, that would help, but I can
I'd be happy
just
to.
ask, what is the PEVK ratio? You had that like color
Yeah,
coded.
the PVK
I
ratio.
did not
So,
understand
okay,
that.
so if you remember, the exons were colored sort of green to pink. It was some sort of scale, right?
Yeah.
And when you were green, it meant that that particular exons was entirely made up of P's, E's, V's, and K's. These are actually the names of the amino acids, right, that are in that are being expressed. And those amino acids can tell us something about the composition. So there's lots of repetition in some of them. They're almost all P's, E's, V's, and K's. Turns out the E is glutamate, and glutamate can actually change the charge of your Titan, of that particular part of Titan. And in changing the charge, it can impact the way in which the PVK interacts with actin, and that could potentially influence its stiffness. So I'm interested in how the composition, that is the P's, E's, V's, and K's, actually impact the property of that region of Titan. Does that help?
So what does a low PEVK ratio mean?
Honestly, I don't know, right?
Okay.
But it might be more that some of these regions are more interactive with actin, and so they have lots of E's in there. And some of them have less of those E's in there, and so they're less interactive with actin. Does that make sense?
Yeah, so I guess it's just a ratio of the 4 different components.
Yes, and there can be some other things in there, but we're learning in steps. Is that we're kind of thinking about the P's, E's, V's, and K's.
Thank you.
Yes. Other thoughts, questions?
What's sort of the relative contribution of Titan versus like matrix components versus like inherent properties of the contractile components that impact stiffness of the muscle?
No, this is a good question. You're probably not going to be super satisfied with this. First of all, historically we thought the tendon was all the elasticity. And what we then realized is that no, actually muscles can recoil too. They can store and recover elastic energy. And from our research on the frogs, it was almost entirely the muscle because there wasn't much tendon associated with it. So what I'm going to tell you is that it's probably dependent on the structure of the muscle. Is that in some cases you might see more of the muscle kind of contributing than the tendon because you don't have much tendon. In other cases, like you think of cursorial mammals running with really long tendons, the tendons might be doing most of the work, and maybe the muscles don't have as much contribution, viscoelastic properties that are contributing to the work of the muscle. And then others, they have this equal contribution. So my suspicion is that it depends if that's, which is not a very satisfying answer. But what I would encourage us to do when we start asking questions is really thinking about how can I test the contribution of the whole muscle tendon unit, and then really think about what's the muscle doing. Is it also, storing and recovering elastic energy? Is it dissipating energy? And what is the tendon doing? And we need to think about both structures. I hope that answers your question some.
Yeah, it does. I would just ask, how would we go about determining those relative contributions?
No, this is a good question, and honestly, where I was hoping that we would go is that how do we test these things? So I think the first thing that we can do is let's start with what we know how to do. We know how to do these isolated muscle experiments, right? And so kind of going back to, I think it was your comment earlier, is that we need to start doing some of these work loops or sort of simulated sort of length changes, stretch shortening cycles where we know we can actually change the length of the muscle and look at how force changes with length. So in my lab, we do stretch experiments where we can characterize, what's the impact of a stretch on the muscle. So I need to think about I'm gonna, the Titan isoform, and I need to think about the muscle property there. Then you might be able to think about, okay, what is Titan or tendon doing in my system? If you're in a system that has a big long tendon, then maybe you also have to measure those tendon properties. And in some labs, which some of you might be part of, you might actually be studying the properties of the entire muscle tendon unit. Things to know is that we all can't do everything, right? But we can start to ask those questions and say, okay, how can I do this systematically? Can I characterize the muscle? Can I characterize the tendon? Can I think about this as an entire unit?
Thank you.
Yeah. Other thoughts, questions, comments before we leave?
Sorry, it seems that the actually vertebrates just use single titin mucus, but we have two different and independent protein in invertebrates that they are acting as titin, like in Drosophila, for example. So my question is that, so does having two separate or independently tunable protein give invertebrates some mechanical advantages in force generation in comparison with us or vertebrates?
This is an excellent question. I'm going to tell you that I don't work on invertebrates, but I agree with your assessment. So just so everybody it knows what it had just talking about is that in invertebrates, like your insects, like the Drosophila, there's more than one gene that expresses a titin-like molecule. And in that, you could have different isoforms of that. So you can imagine that you just have more variability that you can play with. You can have, it's not just one gene. In vertebrates, we only have one gene, and we can undergo all this alternative splicing. My suspicion is that there's just different ways to get variation. In vertebrates, we only have one gene, but we can undergo lots of different alternative splicing. In invertebrates, you seem to have more variation by expressing different genes. I think of myosins, right? We think of myosin isoforms as being associated with all our different fast twitch muscles, slow twitch muscles, but it has a family of genes as opposed to having one gene. So those are two different ways that you can get diversity. All right, I know I'm going over time, so I apologize. I just want to make sure that everybody feels like they've gotten their questions answered, at least a little bit. I appreciate you guys coming today. I know it was the first workshop. I had you guys doing a lot of things. I hope you learned something today. So good luck this summer.